DAAN GENE RNA and DNA Purification Kit. The sample preparation area is mainly used for sample processing, nucleic acid extraction and sampling, which is the area directly facing the infectious samples. Configure the inhibitor remover, deionized solution and carrier RNA dry powder in the kit according to the instructions. Prepare 1.5 milliliters sterile centrifugation tube and add 50 microliters protease K, 200 microliters lysis solution, and 4 microliters carrier RNA to the sterile centrifuge tube. Cover the tube tightly, swirl it for 15 seconds, and mix it well. Place the centrifugal tube in a 72 degrees metal bath and treat it at a constant temperature for 10 minutes. Meanwhile, preheat the eluent in 72 degrees. Add 250 microliters of anhydrous ethanol to the centrifuge tube after the warm bath and close the tube lid. Swirl it for 15 seconds. Cover the tube tightly, swirl it for 15 seconds, and mix it well. Pipette the mixture into the spin column. Conduct centrifugation for 1 minute at 12,000 rounds per minute. After centrifugation, install the spin column into a new collection tube. Add 500 microliters of inhibitor remover to the spin column. Conduct centrifugation at 12,000 rounds per minute and at room temperature for 1 minute. After centrifugation, install the spin column into a new collection tube. Add 500 microliters of deionized solution to the spin column. Conduct centrifugation at 12,000 rounds per minute at room temperature for 1 minute. After centrifugation, install the spin column into a new collection tube. Add 500 microliters of deionized solution to the spin column again. Conduct centrifugation at 12,000 rounds per minute at room temperature for 1 minute. After centrifugation, install the spin column into a new collection tube. Centrifuge the collection tube with the spin column at room temperature at 14,000 rounds per minute for 3 minutes to remove residual ethanol. Remove the spin column and place it in a new 1.5 ml sterile centrifugation tube. Open the cover of the column. Place it in 72 degrees metal bath for 2 minutes. Carefully add 72 degrees preheated eluent 50 microliters directly above the membrane of the spin column. Cover it tightly and let it stand at room temperature for 1 minute. Conduct centrifugation at 14,000 rounds per minute at room temperature for 1 minute. After centrifugation, the nucleic acid solution in the centrifuge tube can be used for PCR amplification experiment. It is recommended to use immediately. If necessary, please store it at -20 degrees.